speak this session, Professor Andy Pickford. He's from the University of Portsmouth, sorry, that was bad. And he's going to talk about the enzyme research that they're currently doing in Portsmouth. So over to Andy.
Thanks very much, Ria. Oh, I think okay, just move across. Thanks, Rob. Okay, yeah, thank you to Rob and Ria for inviting me to talk to you today. So yes, I'm Andy Pickford. I'm the director of the Centre for Enzyme Innovation at the University of Portsmouth. I'm a biochemist, so it's a little different, and I have to say I'm really out of my comfort zone, but it's a really exciting project that we're getting involved in, that we've just started with Rob and Ria in the last month. So as I said, I'm a biochemist, and actually my inspiration comes from pictures like this, which I know is a million miles away from the ocean. But what I see when I look here is polymers. Polymers, natural polymers. This image is stuffed full of cellulose, of lignin, of cutin, of starch even. And these are all natural polymers that support life. Although our eye is drawn to the green in this picture, what's actually really interesting from a recycling point of view is the brown stuff, the leaf litter at the bottom. That leaf litter is going to be decomposed by bacteria and fungi, and all those polymers are going to be broken up into their building blocks, monomers, which can then be returned into the circle of life to make new materials. So nature basically recycles everything and doesn't waste anything. And the things that are responsible for that recycling are what we call enzymes. They're biological catalysts. So for every naturally occurring polymer, there's a naturally occurring enzyme or enzymes that can break those polymers down into their building blocks for reuse. So on the top left here is a very complicated structure, and this is cutin, which forms the waxy surface that you find on leaves, which makes them waterproof. And cutin is a polyester, albeit a very, very complicated structure. I've highlighted here in circles the ester bonds that link it all together. And you can see the similarity then with the synthetic polymer polyester, like polyethylene terephthalate or PET, which we use, of course, to make single-use drinking bottles and food containers. This molecule is actually a lot simpler than cutin, but contains the same ester bonds, which could be targeted by enzymes like cutinases. Similarly, we find other polymers, naturally occurring polymers like protein, which can be broken down by proteases, and you see the similarity in the bonds there, the amide bonds that we also find in polyamides like nylon 6,6. So the question is, can we use these enzymes to break down synthetic polymers, plastics, that have similar bonds? So our ultimate objective would be to do something like this. We take our waste plastic, which is long polymer chains, as hopefully we know, of these building blocks, polymers that are linked together by certain chemical bonds. And we use enzymes to break those down into a soup of the building blocks. We separate those building blocks by some downstream chemistry, and then we reuse them. So either to remake the same polymer, or we can upcycle them into something more perhaps useful or of higher value. For example, this compound, terephthalic acid, you can turn into vanillin, or the vanilla flavoring. So basically what we're trying to do is circularize this economy so that we almost decouple, if you like, from oil and gas, so that we retain the carbon within the cycle. So in order to do that, we need enzymes that are really efficient and really tough, so that they can withstand the industrial processes. Just a little bit about what we need to do. We need to engineer enzymes. There's a lot of information on this slide, but basically enzymes are proteins, and proteins are themselves polymers. They're biopolymers, and they're made up of long chains of amino acids, which is what this jumbled alphabet represents here. And that is encoded by our DNA, or the DNA in whatever organism which makes that enzyme. So if we want to change the enzyme, we identify the kind of business end that we're interested in. We make changes there by changing the amino acid sequence. And actually what we do is we go back and change the DNA sequence. We call this structure-guided enzyme engineering, because we need to know the structure of this enzyme. So we first got involved in this area in about 2016, 2017. It was about the time when there was a fascinating article published about an enzyme that was discovered outside a plastics recycling plant in Sakai City in Japan. And the microbiologists there were looking at the organisms which were growing on the plastic and in the soil underneath where the plastic was being stored. And they found this bacterium, which they called Ideonella sakaiensis, that was sticking to the PET water bottles. But not only was it sticking to it, it was actually digesting it. And it was using the plastic as an energy source and as a carbon source. Now, this is kind of remarkable because we've only been making plastic for a number of decades, since the 1950s. So the fact that evolution has worked so quickly that we now have bacteria that are able to use our plastic as a food source is quite remarkable. So those bacteria producing enzymes, the famous one is called PETase, which you may have heard of, which breaks down the PET into kind of a cluster of related molecules. And then another enzyme called mhetase finishes the job off and produces the two monomers, TPA and ethylene glycol. And this was published in Science in 2016. We picked up this story and we thought, well, what if we can make PETase better? Can we make it more useful perhaps for industry? So we started to try to understand how it works. And in doing kind of standard laboratory tests, actually we accidentally made it 20% better when we were actually trying to knock out the activity and try to make it rubbish. So that kind of hit the headlines, and it kind of went viral. Kids term viral, not my term viral. And it caused a great deal of interest and impact. And off the back of that, we set up the Centre for Enzyme Innovation. And our thinking was that, well, clearly we could set up a research and innovation pipeline where we go out and we try and discover microorganisms and enzymes that might be of use for breaking down plastic. We would then engineer those to make them stronger and faster, more robust and useful for industry. We also engineer the way that we make the enzymes, because it's all very well and good having a technological solution. But if that technological solution is too expensive for anybody to use it, you're not going to succeed. So the enzymes need to be cheap as well. We then look at different ways of deploying them, and ultimately we apply them in the lab in a process where we break down the plastic, we recover the building blocks, the monomers, and then we try and put them back together into a plastic which is indistinguishable from one that you might make from original fossil fuel-based resources. All the time we're working with industry because we're very conscious that whatever we do has to have real-world significance. So we were awarded nearly £6 million from Research England in 2019 to set up a centre, and then we had further funding from local government to expand a couple of years later during lockdown. This slide, a lot going on here, but I just want to show what you can do by engineering enzymes. And this graph basically shows how enzymes perform if you try and use them at higher concentration, try and use more of them. And actually the natural PETase is really rubbish if you put too much into your soup of plastic. It really suffers. If you engineer, you can engineer this property out by making the enzyme more tough, more thermostable. And this one you can see hot PETase massively outperforms the original one. This is something we published earlier this year. Another way of engineering enzymes is to couple them together. I'll try and stop this because it makes everyone dizzy. And in this case, we just basically stapled the PETase and the mhetase together. Very, very, very simple piece of engineering. And actually that increases the rate that the plastic gets broken down 6 times. So an astonishing improvement, rather unexpected again. what we do, as I said, we're really conscious that what we do has to have real world significance. And so whenever we do our small scale experiments in the lab, we always try and replicate how those enzymes would be used in industry. And this graph over on the left here, we call this a heat plot, which is just testing lots of different kind of enzymes coupled to little, what we call little anchoring domains, like this little foot here, at different temperatures. And we're trying to find, what might be a great combination. And this particular hotspot here, you can see looks really, really good. This was at a tiny little scale on a couple of mils. But unfortunately, when we scale that up then to a kind of system that would mimic what you'd use in industry, where you've got about 200 grams per litre of plastic in a slurry, actually you don't see much difference at all, which is disappointing, but really, really important to make sure that what we do is industrially relevant and important enough to get the front cover of the journal. We also screen many, many different enzymes. There's lots of information here, but basically this is like a tree of life in the middle here. Screening from lots of related or distantly related bacteria or fungi, looking for enzymes which might have the properties that we need. In this case, we are looking for enzymes which can tackle crystalline plastics, so really, really tough PET, and we found a few which look really promising. We worked with Google DeepMind on this project. You may have heard of DeepMind. These are the AI guys, the AI masters. And they just developed a system for predicting what these proteins, what these enzymes look like, which is determining those experimentally. Each enzyme takes about 3 months. They did 75 for us in a weekend. So you can see how AI just is transformational for the kind of work we do. Now, as I said, industrial relevance is of course actually vitally important, and biorecycling with enzymes is getting to the point where it is being adopted in industry. This French company, Carbios, who were a lot of pioneers in this work, they're opening a pilot plant they have a pilot plant open. They're opening a 50,000 ton per annum plant to deal with biorecycling PET sometime in 2025. So there's a lot of investment in this area. So that's about what we do. We're just starting out on a project now with Rob and Ria, funded by OSRL, which we're really excited about, to look again at real world significance. And you've seen images like this already at the Express Pearl disaster, and there are people in the room who've had first-hand experience of this. But our question is, well, can our enzymes that we have tackle the nurdles that have been collected from the environment? And in order to break them down into those building blocks and do something useful with them. Now, unfortunately, the enzymes that we have developed to date won't do that. And the reason they won't do that is that enzymes are very, very picky about the types of polymers that they will break down. So PETase will break down PET, which this is a model of it here. The red there are the oxygens in the ester bonds. And despite the very similar name, polyethylene terephthalate and polyethylene, you can see polyethylene is a very, very different monomer. Polymer. It's very greasy in that, as far as enzymes are concerned, it all looks the same all the way along the chain, and there's not really anything for it to grab hold of. So Hayden, who's here in the audience here, who started his MRes with us, funded by OSRL, has done some analysis of nurdles collected from the Express Pearl disaster, and he's found that what's been collected from the beach cleaner is actually a mixture of low density and high density polyethylene. So LDPE is the kind of polyethylene you'd use for plastic bags. HDPE is the type you'd use, for example, for milk jugs. So there's different applications, but we couldn't find any PET. PET is a lot tougher. It melts at around about 260 degrees, whereas these two you will see about 130, 140 degrees. So we haven't got any PET. So PETase won't work. So is there an enzyme then that can break down PE into its monomers? Well, possibly yes. There has been a report very recently of enzymes in the saliva of waxworms. Now, bear with me with this, it's slightly crazy. This is a story, Professor Bertuccini here from Spain. She is a keen beekeeper, and she found that her beehives were getting infested with these worms from this moth. And she collected these waxworms, put them in a plastic bag, just to dispose of them, and then noticed that actually the worms were chewing holes in the bag. This was a polythene bag. Being a good scientist, she started to look into this, and she found that if you sample the saliva from the worms, which I can't imagine how you do that, but with a capillary, I think, you can then actually just put the saliva on the polythene, and it starts to break it down. Not very fast, and not to a great extent, but there are enzymes in the saliva of these worms which will break down polythene. Now, it doesn't do it in the same way that PETase does. It seems to oxidize it and cause it to fragment into a soup of different chemicals. But if we're just trying to get rid of the plastic spill, that's probably not a problem if we're not necessarily looking to do something with the product. So these enzymes are inefficient. We expect that anyway, because they're natural enzymes, and the waxworms would normally eat wax, of course, from the beeswax, not polythene. But we think maybe we can engineer them to make them better. So that is the ambition behind Hayden's MRES project. We started this project just this month, as I said, funded by OSRL. And the intention is to genetically engineer yeast so that we can make these waxworm enzymes. You might think, why would you do that? Well, you simply can't get enough waxworm saliva to make this an industrially relevant process. But what we can do is we can use yeast. We can program the yeast with the DNA, which encodes those waxworm enzymes, and basically use fermentation to make the enzymes that we need. We're then going to explore the enzymes' ability to break down that polyethylene, both the LDPE and the HDPE, and try and figure out what the limitations are. Why don't they work as well as, for example, the PETase enzymes do, which I should have said. PETase can completely, or PETase-like enzyme, completely depolymerize PET back into its building blocks, so there's no polymer left. And that's our ambition for PE. And then we're going to start engineering those enzymes to try and make them more efficient. So that's it from me. Again, thank you to OSRL for the funding, and just to highlight the people involved in this, including Louise Farrand, our Innovation Fellow, who's at the back of the room there, Hayden, Victoria Bowers, and Bruce Lichtenstein are two of our Senior Research Fellows in CEI, and Elaine and Kerry provide our specialist technical support. So thank you very much.
Thank you, Andy. Any questions?
Yes, Ken Lee from Canada with Fisheries and Oceans Canada and Natural Resources Canada. I'm just wondering, can you comment on the cost of doing something like this in terms of genetically engineering these enzymes and what the cost would be to try and use it as a remediation?
So the cost in terms of genetic engineering is a one-off. So that's the kind of research and development cost. It's a very well-established process. The changing the DNA is very well established and pretty cheap these days. The costing is really in time, because it's a very iterative process. You make changes, you use as I try to indicate in our thing here, it's an iterative process. You make subtle changes, you see what works, you take the best of those particular panel of enzymes and you make more changes. So it is quite time consuming. There's a process called directed evolution, where we try and speed up the evolutionary process, but do it in a lab environment, which massively increases the speed at which this can happen. So it can be done, in maybe a year to get really good improved enzymes. It's difficult to put a fixed cost on it because it depends on the difficulty of the challenge. Once you've got the enzyme, it's really, really cheap to produce. So the expensive part, actually, when you look at, for example, PET recycling, the expensive part is actually, well, the most expensive part is buying the PET flake from the recyclers in the first place. That's by far the most expensive part. Pre-treating it so that it's suitable for enzyme digestion is also expensive. And then recovery of the monomers is quite expensive as well because you have to do distillation. But the actual putting the plastic into a reactor with the enzyme and depolymerizing it, that is the cheapest part. So in terms of if you just want to get rid of plastic and turn it into some kind of soup that microbes might feed off, that's actually really cheap.
Okay. Thank you.
Brian Mitchell, Run Consulting. I guess the PET is a pure polymer, but a lot of plastics are fed with flame retardants, particularly brominated flame retardants. So is that folded into the picture of this research?
It is, yeah. We're very conscious that there are a whole variety of additives. There will, flame retardants. There will be catalysts there, of course, from the original polymerization. And what you need to do, when we work in our controlled environment, of course, we capture those in some downstream processing through, for example, activated charcoal or something like that. That's something that we need to be conscious of. If we do this in an open environment, then we don't have that ability to capture those potential toxins.
Yeah.
So that would need to be set into some kind of risk assessment of how you would actually do this in the field. If at all you do it in the field, or do you bring the collected Nurdles back and you do it do the deconstruction in a contained environment?
Actually, do the Nurdles contain the flame retardants, for example, or are they raw material?
If you're using, for example, PET to make polyester textiles, then I think those are typically added afterwards, maybe in a melt extrusion process or even sprayed on afterwards. So I suspect they're not in the original Nurdles.
Okay, thank you very much.
Any more for anyone?
Hello, Lizette Wood from Chemistry Laboratory. I noticed that both of the plastics that you're working with are the ones that are actually a little bit easier to recycle. Anyways, besides the additives, which is a good point, but there's certainly some other plastics that are not very easy to recycle. Is your team intending to also look at those? It seems right now it's reactive to finding something that will break down the plastic and perfecting that, but is there intentions to have some active looking at things like polystyrene or?
You're right. PET or polyesters are really the low-hanging fruit. The bonds are quite weak in comparison to polyolefins.
Yeah.
But we do have active programs on polyamides, nylon, on polyurethane as well. This is the first project we've been doing with a saturated carbon backbone. So we're not doing anything on polypropylene, for instance. We're not yet working on polystyrene, but we have collaborators who are looking at that. My gut feeling is that biological recycling will not be appropriate for every single polymer type. But until we actually try it out, we won't know. And there will be chemical recycling processes that are more appropriate in some cases. What enzymes do have going for them is that they're very, very tolerant of contaminants, which of course a lot of chemical processes are not necessarily that tolerant. And chemical catalysts can very easily get fouled by off-target polymers and contaminants. So enzymes in general, don't give a monkeys about what else is in there, because if you think about it, in nature, they're working in a very, very mixed environment anyway, of a soup of all sorts of stuff, but they focus in on the particular target. So in general, enzymes are tolerant of most of the contaminants, but most proteins stick very non-specifically to polystyrene. So polystyrene may well be a real problem for enzymes, but others perhaps not so. Sorry, very long-winded answer.
Bronwyn Lee from Oils for Response. I'm just wondering, in terms of kind of other applications, can these enzymes, are they only used in controlled environments or for kind of ocean cleanups, like big garbage patch kind of things? Can they be applied in a situation like that, or is it just a controlled?
It's a really interesting question, and it's something we've kind of toyed with and debated with quite a lot. So what we're not looking to do is just scatter enzymes into the sea. Partly because it would just fail. The enzymes would be diluted to such an infinite level that they just wouldn't do anything, because they're just diffusing around. That said, what might be possible is, once you've collected, from the garbage from the Pacific or something, it might be possible to break it down on the ship. If you had a bioreactor under suitable conditions, if you had enzymes which could perhaps work with in saline salt water, so you didn't have to take fresh water out with you. So those kind of things might be possible, and that would avoid then having to ship, and all the greenhouse gas emissions of having to drag all that plastic back to the shore again. So maybe it's, we're a long way off that. So.
I think we've got time for one more.
This is really quick. Hey, Kelly McFarland, to piggyback on what you were just saying, you should also keep in mind just the lifespan of these enzymes too. So that we know the reason why we can't just like spray them on the ocean, because they would degrade quite rapidly.
That's right, they would. They would not last very long. We do engineer them to be more robust, but they will suffer UV damage. They will be broken up they'll basically be food for microbes in the sea, because they're just protein. So yeah, putting enzymes into the ocean is not an option. You're absolutely.
Great, thank you very much, Andy. I think that concludes this session. So yeah, thank you very much to all